this week?
Streaming.
Why does it look like it? Oh. Alright, catch you guys tomorrow.
Yeah.
Bye. 2 viewers. Oh, I'm excited. Are you excited? 2 viewers. Oh, don't turn those off, please.
Oh, you use those?
Yeah. I look great in the background.
They start coming on again.
I'm just gonna sing that the whole time. Hello. What y'all up to? Oh my God, Inception. I wonder if I can move this. Oh, I can. Hey, Christopher, what's up? Display. Yes. Hello, Anto. Joseph, hello. Be right back. Yeah, we coo, Jill. We coo. We coo. See you tomorrow. See you tomorrow. How are people's days? Happy Tuesday. What's up, Tom? Glad to see you guys here. Chris had a long weekend, well rested. That is super good to hear. I'm so glad. Tuesdays are always happy. Because we're online. Duh. Everyone have their beverages. This is my beverage of choice. Via Coco, you know, sponsor me. I'll do whatever you want. Be your spokesperson. Yeah. I know Josiah prefers beer, but I get like Asianglow. I don't know if you guys know what Asianglow is. It's a genetic thing. It's like 98% of Asians get like this glowing like red face, and then like yeah, because in Asian cultures we can't break down the enzyme ALDH2 in alcohol, so we have like this really bad allergic reaction. It sucks. So if I drink like a beer, I get really red. Yeah, it sucks, Chris. David asks, are those XX chromosomes, or are you a fan of the band of the XX? Actually, I'm glad you asked. This is a sweatshirt from a like really cool street artist named Kaws, Kaws, and he did like a collaboration with Uniqlo. So like he likes putting little X's on people like things eyes. So like you could see it's like a Sesame Street collaboration, which is pretty cool. Yeah. So, and I guess I'm feminine. And I do like the XX. Yeah. So all the above. Yeah, David. I get alcoholic flush reaction. And I'm also lactose intolerant. I'm like a genetic Asian mess. But that's why we're all here. to make ourselves better. Yeah. Yeah. Okay, so it is 5:02 and I'm ready to rip because I really want to go watch a Sharks game tonight. Go Sharks. So let's talk about sequencing and reactions. All right, so week 4. We just hit the middle point of our class. There's only 4 more weeks left out of the 8-week course, and after this video, we should be at the end of teaching. So everything prior to this video, we'll all be using for the next 4 weeks to make something beautiful, which is pretty cool. So everything that you learn from mini prepping to gel electrophoresis to analyzing sequences will all be applied in the next 4 weeks, and which is very exciting. So yeah, I'm pretty excited. Anyways, so let's talk about sequencing. Now, I absolutely love talking about bioinformatics, about DNA, about reading DNA. I think big data is super sexy, and I think if I weren't a genetic engineer, I would have gone into genetic counseling, because looking at the DNA is super amazing because you can manipulate it in so many ways and find out so much information about it. So I'm going to give you a little bit of background about just like sequencing in general, and then we'll dive into the sequencing ourselves. So Sanger sequencing. Have you guys heard about Sanger sequencing? I'm so parched. Via Coco, sponsor me. Well, Sanger sequencing is a sequencing platform. So, and it was one of the first types of sequencing to come about. Before, like way back in the dark ages, we didn't know what DNA was or how to read it, or the sequence of A's, T's, C's, G's that make up all the proteins, right? And so Sanger sequencing was actually born 40 years ago, and it was really ambitious to sequence the whole human genome, because it's so large. I forget how large is the human genome. Genome sequence. Oh my god, I really should know this. How many base pairs is a human genome? The human genome contains 3 billion pairs. Power of Google. And it's a ton, it's a lot. And Sanger sequencing was the first ever to be able to tackle this type of project, to be able to call out every single base pair, A's, T's, C's, and G's, which is pretty cool. So the human genome was sequenced between 1990 all the way to 2003. So it took about 13 years to actually be sequenced, and it took millions and millions of dollars to be able to sequence one person's human genome. So as in this class, I like to reiterate that there is no such thing as a model organism. Right now, the human genome sequenced comes from one man. And, there are some mutations in humans, and it doesn't encompass everything. So if you go on NCBI and open the human sequence, I believe it's from that one guy, which is not super great. But there are things like, there are companies like 23andMe, and like other genetics companies that's working on creating a very large human sequence database. It's not open sourced, and we don't have access to it because of laws and protection laws and such, but these kinds of projects are going on. So I'm just really interested in the human genome because number one, we're humans, and number two, there's so many base pairs. Like I said, it was 3 billion base pairs, so it's just a lot to process. T. Williams asks, what is your opinion on junk DNA? Do you think it all has function and we just don't understand it yet? So yes, I believe that. I believe that is the yes. I believe so. So like I said in my previous video, there are introns and exons. Oh wait, is that this class? Nope, it wasn't this class. Sorry, my bad. I was talking about it in the 101 class. Yeah, so I was talking about when DNA translates through the central dogma of biology from DNA to RNA to protein, that there are introns and exons. And for the longest time, we believed that introns were kept in the genome and processed to proteins, and exons were exiled and excommunicated and exsliced out of the protein sequence and was complete junk. Now, we don't know the complete function of every single sequence in the human genome. So it's because it's just not been studied. We've just recently started amassing all these people's genetic information to be, saying these people had breast cancer, what kind of gene creates this, or is this even genetic? And so they would take all the breast cancer diagnoses and sequence people's genomes and figure out, oh my gosh, there's actually a single point mutation inside of the 3 billion base pairs that humans have, and they're like, oh, we deem that this one particular mutation in one of the particular genes causes breast cancer. So we're still mapping out the human genome, and it might take a while because we don't know how it interacts with each other, if there's any sorts of epigenetic or environmental sources that we might be missing. So a lot of the genome is very unknown function-wise. So it's up to us to continue the research to, look at gene interaction, protein interaction, and things like that. Okay. So let's talk about today's sequencing. So I mentioned Sanger sequencing. Now Sanger sequencing, like I said, came about 40 years ago. And sequencing since then has been going through the roof. I think we're hitting a second golden age of biology to be able to expedite and create things on a much faster and cheaper scale. So there is another company called Illumina that introduced the Illumina sequencer. And this is considered a a short read sequencer. And it's part of this thing called a next-gen sequencer. So Illumina has really rapidly changed the face of genomics in the past maybe 10 years. For example, the human genome to sequence one human genome was $1 million. And I think it took about several weeks to completely sequence the whole entire thing through Sanger sequencing. Now with Illumina, with the HiSeq or the MiSeq, it's much faster, and you can sequence a whole human genome, the same whole human genome, in much less time and only for $4,000, which is incredible, right? This is the cost of 2013. I imagine it's much cheaper today to sequence a human genome. So it's incredible. 20 years ago, we were pulling our hair, oh my God, it takes so long to process and produce, one whole human genome, and we could do it on a scale of $4,000 in a couple weeks. And I think it'll get much faster. There are other competitive outlets such as Twist Biosciences that's making, sequencing faster, and there's just like so many things out there. For example, like the Oxford Nanopore. The Oxford Nanopore reads DNA, is a tiny next-gen sequencer, and it's literally about this big. And you can literally just plug it into your laptop and drop a few library prep it, and it'll just immediately run. And within like 30 to 40 minutes, you can get actual DNA sequencing. And I think the flow cell itself, it only costs $1,000 to sequence. So it's just gonna be really, really fast. It's gonna be super accurate, and like there are tons of people within my community, in the biohacking community, that's literally just like barcoding everything, sequencing everything, just to see like what we get, and it's just we can do a lot with some of the data. Buzzwordy09 says, did you ever try the Nanopore, and is it any worth? We have tried the Nanopore. We actually have a Nanopore in-house, and we tried to use it for brewing yeast, trying to sequence some of the novelty, like proprietary brewer's yeast. And in my opinion, it was really difficult to use. The Oxford Nanopore is very, very uptight about their proprietary flow cells and also the technology, because they don't want you to reverse engineer it. So they do tell you to sign a contract to make sure that you return the flow cells after you use them, and they're not allowed to be sold on the internet. So that was kind of frustrating, because we're just like, oh, open source everything, but we can't. And then on top of that, it was really difficult to follow along with the protocols. It was very, very tricky. And then we tried to get a little bit of help from their IT department, and they were not super forgiving or responded to any of us. So I think Peter, I, and Josiah, we've kind of finagled around with it for several days, and we just got like really terrible sequencing. It could be our library prep. Maybe we're just not very adept at it, but I haven't had too many biohackers in the community that have worked with the Nanopore like super well. And I think the recovery is not very good, so it's not super effective and accurate yet. But we send our sequencing out to like GeneWiz, and they do a really great job, because they're an actual company that has Illumina sequencers, and they know how to library prep everything. So the source of error is very minimal in that amount. And also the Illumina produces some really high quality reads. So, and it's also like $7. Like, how can you go wrong with like $7? William asks, how accurate was a Nanopore versus Illumina? The covery, like there's something called the covery, and we'll go over that today. The query and the covery are kind of very similar, to where you can see how accurate things are. So what I'm going to go over today, I'm going to show you how accurate some of these methodologies are. Illumina versus Nanopore. I know that Illumina is much more accurate than the Nanopore based on the technology alone. So I can't tell you like how far of a difference. But okay. So we talked about the Illumina. Let's talk about how to submit your samples. So I don't know how many of you have successfully isolated or mini prepped your DNA separately from your cells, and then right after that you had to run it through the gel electrophoresis. And I'm sorry, the document is not up yet. Josiah is traveling to China right now and barely had the time to even like look at it and write a few words out. But we will be coming up with a document shortly to show you like what are positive and negative results for like the gel electrophoresis. I'm not 100% sure what each thing that you isolated reads. I think it's pretty high up there, like maybe like 10,000 base pairs or so. I'm not 100% sure, so I'll get back to you on that in the Google Classroom. But anyways, so once you get to that step of gel electrophoresis, confirming that you have products, then you can go ahead and pack that up and then send it off to GeneWiz. So people that live in the United States have access to GeneWiz. People that are not residents of the United States, I'm sorry, you need to find another sequencing company that can potentially sequence it for you. Or if you can't find a sequencing company in your country, you can just ask me, email me. I will send you some of our results that we've gotten back in the past, and you can analyze it with us. So, yes. All right. And yes, so submitting the sample, you can find the document in our Google Classroom. I can go over it really quickly, but essentially you make a account with GeneWiz, and then you set up your account, and then once you isolate it and purify the DNA, then you put it in a tube and you parafilm it, and then you put it in a bubbler. Oh, and the amount of DNA that you usually ship is like 20 microliters, which is completely fine for this reaction. They only maybe use like up to 3 microliters, I know, but in this case, we'll send them 20 just to be safe. We don't want them to be like, you didn't give us enough DNA. So anyways, wrap that, put that in the mailer, mail it out, and it's okay to ship it at room temperature. Like I said, DNA does not degrade for like 6 months, like outside a refrigerator. So you're completely fine. It'll get there. It'll arrive there. And then for sequencing, you choose the T7, T as in Tommy 7 primer from the drop-down list. And it should be on its way, and you should get your sequencing results within a week. Pretty quick. It's pretty quick. Okay. So, today I will show you how to read your sequences. It's important. It's great. It's super important. So I'm going to do something where... Nope. Display capture. Okay. My bad. I'm sorry, this is crazy. Okay. So let's take a look at some chromatograms. Well, before I do that, GeneWiz will send you 2 files. They will send you a chromatogram and also, I believe, an AB1 file. For the purposes of this portion, we're going to be looking at the chromatogram. So I've already opened it up. I'm going to expand it as much as I can. All right. So you're going to open up the chromatogram. Now this is an example, so don't be alarmed. It's okay. I'm just going to show you kind of the key highlights to what you should be extracting from the chromatogram. So you're going to open it up to this, and you can see that there's a standard, an auto-scaled, a raw data, and the chromatogram data. So the chromatogram data is just the raw files, A's, C's, T's, and G's. but we want to look at the auto scale. Now you could look at the standard scale, but the auto scaled works pretty well for us. So you can see that, let's see, can I scale? You're going to see like something very similar to this. You could see that it goes from 0 to over 1,200 in base pairs. And that's probably the whole entire fragment of DNA that you went to get sequenced. So let's try and expand this out a little bit. You can also move the scale on the bottom here to 1x, or you can move up and down the scale. Woo! Which is a really cool feature. And then up here you can see the base calls. So when things are sequenced, it calls out a single base pair. And this is your sequence to your data. So for example, A's are green, T's are red, C's are blue, and G is black. So let's go to a region that's pretty clear and pretty concise. I'm just going to scale it down so you guys can see really well. Maybe I could expand this as well. All right. So this is your beautiful, beautiful data that you will get soon. And this, for example, this region right here is it's like ATCA, and you can see that in the A position that's called, it is green. And you see this really, really high peak here. There's like peaks and valleys. This is when each base pair is called. If you show quantity values, which is this button right here on the bottom, you can see every time the base pair is called how much the quality was. Now remember when I was talking about like, oh, the covery region and stuff like that and how accurate it is. So I believe GeneWiz uses Illumina sequencers, and yeah, we can with confidence say that in this peak, this really, really sharp single peak, the call was an A. And you could check from the data. It says quality 59. Higher the quality, the more confident you are. Now let me show you a place where the quality might not be so prominent. Okay. I guess there's not very many within the sequence. So usually when you open up your sequence, there's lots of ends, and ends is the one that's indeterminate. The sequencer and the program couldn't distinguish what base call it is, so that's why they show up as ends. I don't see any here within the sequence. It seems to be a pretty good clear-cut sequence, but let's see right here, for example. So there's an N right here in the 997th base pair call position. And you can see the quality is 0 because it was very unsure what it was. So when you read over this, you can actually go over the sequence and correct for it. Because if you input this sequence into, like, let's say Ncbi Blast, it'll be like, what is this? I don't know what this is. N, I have no idea. So what you can do is you can go into the chromatogram and go into the actual data and see that this was an A calling, so that called it pretty correctly. And then there is a blue peak and a green peak here. And it looks like the green peak kind of overlaps with the blue peak. I'm sorry if you're colorblind. But it looks like the blue peak is the highest here. Now, it may not be the most quality call because there are some factors that can lead to calls like this, maybe contamination or just like a simple mutation, or it just wasn't sequenced properly. So, but it's okay. Like, we can see that the blue peak here is a C. So what we can do, I don't know if you could do it here. Nope, you can't do it here. What we can do is remember the 997th position, and you can not do that. Can you edit this? So I forgot to tell you, this is actually on, this whole thing that I'm opening up is downloaded from SnapGene Viewer. So there's regular SnapGene, which you have to pay for and subscribe to, but SnapGene Viewer, and I'll definitely post the link later, is a free site that you can just use. But I believe SnapGene Viewer, the free one, doesn't allow you to edit within the documents itself. So to bypass this, you can copy the sequence. So we can go back to the chromatogram data, which is literally the same thing, and copy this whole thing. Copy. Oopsie. Well, no. And paste it here into a Word document or like a notepad. And remember, it was like the 997th position. I mean, I guess you can count all the way in. I really don't know where it is. But like hypothetically, let's say this N was a 997th position. You can delete the N and then put in that C, which we called out. So once you go back and fix all the N's within the sequence, then with the accurate human eye base calls, then you can copy this whole thing and put it in Ncbi Blast, which I will show you next. I want to go over a little bit more of this particular graph. As you could see at the beginnings, there were lots of N calls, and towards the end there were more chaotic N calls towards the end. This is because the sequencer kind of gets chaotic at the beginning and the ends, only because the ends of the base calls are just like kind of like junk slash could be like some primer activity. So that's why it looks kind of chaotic here. I generally don't really care about the beginning and the ends. Only the middle sequencing is what I usually care about. So if I go back, I don't fix all the ends at the end. I'm just like, okay, at this position at 1046, between that, the sequencing looks pretty decent. But starting from here, it looks like the peaks are pretty well preserved, and looks pretty sharp and looks pretty good. So I usually just copy and paste like between, you know, this 832nd position all the way to like the 28th position, because it looks pretty good in the sequencing here. I'll show you an example of a really terrible sequenced. So this is an example of like a really, really poorly sequenced. Now there are many things that can lead to bad sequencing. It can be like your mini prep that you did, that it may not have yielded the best amount of DNA. It might have yielded in very little DNA, or it might have been contaminated, or any contamination issues from literally putting your purified product to a brand new tube and sealing it up can contaminate it. It could rarely be GeneWiz. They do like a very standardized library prep on their end, and then they feed into the machine. But most likely it's gonna be your fault. Sorry. So this is an example of a really poorly sequenced data. It's okay, the ends here or whatever. I don't really care about the beginning. But look at here, look at the middle. So in the other one, you saw pretty like clear peaks. There are some background noise on the bottom. You could see these like little bumps right here, but the quality of the calls were very, very low. Or the quality of the calls were very, very high to where it completely outcompeted this background noise. Now, if you have a sequence, you could see there's actually lots of background noise, and they're creating like this like really bizarre looking call thing. You can kind of see it more here. So there's lots of overlap down here, and it's getting a little crazy. This is not too bad actually. The sequence is not super bad. But in a bad sequence, it'll look more like this region here, like all over the place, and it's unreadable. And they're all just like ends, which is not super great. So I've shown you the chromatogram. Any questions? Why are the first 25 nucleotides unclear? So usually the polymerase binds to, I believe, the 5' end of the DNA, and then it starts reading. But you have to realize the polymerase used is like this massive protein that just comes in and just like chonk, like just sits on top of the DNA. And it may not read the first like 25 base pairs very correctly. So there's lots of background noise, and usually when I'm designing these sequences to be read and the primers to be like reading the sequence, like the beginning forward primers and the reverse primers, I usually go upstream 20 base pairs from the region that I want to be sequenced, only because I know I can already anticipate that there's going to be some junk DNA that is unreadable. So I usually go 20 base pairs upstream, 20 base pairs downstream to be able to read the region of interest I'm interested in. Okay, so the next thing that I'm going to talk about is the blasting. Oh, the blasting. I'm super excited to tell you about the blasting. All right, so there's this awesome website called Ncbi Blast. Buzzwordy09, do they mail the files to you by snail mail or do they email it? Yeah, so you made your account through GeneWiz and they will immediately email you the results. It's pretty snazzy, actually. I love it. Okay, so let's get back into it. Sorry, I'm not super versatile in this program. I guess this is cool. I'll just make sure I don't. Nah. Sorry for all the background stuff. Figure out a way to use this program. Anyways, so you're gonna go to NCBI, and if you're not familiar with this website, highly recommend you become familiar with it. The entire genomic database of everything imaginable has been sequenced and uploaded here. So all the researchers and academics, all sequences, all reference sequences are found here. You can also find great resources such as papers, mentions of genomes. You can make your own like phylogenetic tree. Like there's so many resources that you can use from the NCBI. So you're going to click here, and you can see there's lots of resources. It's also linked to PubMed, so if you need to find yourself a quick paper, you can just go and search on NCBI. So we're going to go to Ncbi Blast, which Blasty. My bad. Okay. So this is your Ncbi Blast. We're going to go under Nucleotide Blast. And the purpose of Ncbi Blast is to, so let's say you sequenced your plasmid, and you want to check the accuracy against something that has already been checked before. Or maybe you're trying to figure out if I sequenced the correct gene inside of the gene of interest that I'm looking for. You can use this tool as a great way to figure out and confirm that you have the correct sequence. So you can blast it against each other, and then you can align it with each other. So here it asks you for the fastest sequence, or the accession number, or GIs. So remember that we have these like really awkward AB files like chromatogram files. So what you can actually do, going back to the chromatogram file, so this is the sequence, right? This is a sequence of our file, or our gene of interest. You're going to save as. You can see that. You're going to save as, and you're going to, instead of save as type snap gene DNA, you're going to do the FASTA. Now the FASTA is like the raw data. It literally just has A's, C's, T's, and G's, and you're going to save it as the FASTA file. All right, and I'm going to do that again for another sequence that I want to blast it towards. I'm going to do the open human sequence, or human sequence, and it is, where are you? Open. We're going to do that, file and save as, open human, FASTA sequence, okay. Okay, so now we have both of them in FASTA sequence in this raw data form. Okay, so we choose file, and I want to go what my plasmid was, which is this OH2 FASTA format, FA, open. And the second one, I want to choose and blast it towards a known sequence, such as a human, in a FASTA file, open. And then you're just going to blast it. Now it takes a little bit of engine time to run. Oh, I guess it's pretty quick. But usually it runs through different types of databases, just making sure that this is correct. And before I move down and show you, you can look at this score. So you can see the max score and the total score. So the max score is the maximum amount of data of base calls in the open human. And what I actually blasted it towards was my experimental plasmid, and that's the total score. So overall, it got a 99.87 hit. Hit meaning it completely matches each other. So it matches 99.87. If you go down here, you could see this whole sequence all being matched up, which means I can confirm that my plasmid that I went to get sequenced matches completely up with the human sequence that I wanted to match up to. Yay! Yeah! Super exciting stuff. So again, Blast on NCBI is a good resource indicator to show that there are organisms that exist that you might be interested in. Also, so like for example, if I were to insert a gene or delete a gene or substitute a gene, this would look kind of different. It would probably not be 99.87% correct. It would probably be like whatever you deleted from, whatever sequence you deleted and added or mutated would show up on here. And it would show how accurately it matched with the sequence of interest. Okay. Any questions? Buzzwordy09 asks, it should not matter what country you're in. Yeah, so I said at the beginning of the lecture that GeneWiz is only available through the United States. Outside the United States, you're kind of on your own, unfortunately. You can certainly message me and ask me if there's any particular like sequencing site out there outside of the US. I'm not really familiar with all of them, but I can try and help you out as much as I can to keep your costs low. But I know that GeneWiz does it for us. They're very, very cheap in the United States. We go through them all the time. Pretty accurate, pretty fast. It's a week turnaround, so if you can wait that long, that'll be good. Yeah, so, but if you can't, number one, sequence your own, or if you live outside the US and don't want to sequence it or can't find a place, email me. I could give you a sample of our sequencing that we've done prior, and you can just kind of play around with it. Joe Dart. If the plasmid is circular, where do they start reading? Let me see. Let me see. Is there a good? I don't have a great example here. Next class, I can potentially show you a circular plasmid, and I can show, well, hold on, let me see if I could do that. Un momento. File, new DNA. Close all windows. I'm sorry, I don't have a really good example to show you. let's see. Let me do this. Okay, well, I can show you. So this is just the Add-Gene E. coli rapid plasmid. And as you said, it was DNA is circular and it is a circular plasmid. So what constitutes where to begin reading it and where to end reading it in sequencing? So you design these things called primers, and primers are usually 20 to 25 base pairs that you synthetically make yourself. So what you do is you go into the sequencing that you just got back, the sequencing that you're trying to achieve. So prior to all of this stuff, you already take a model organism and then say, like, let's say you want to work with E. coli, which is what we have here as the map. And you're like, okay, I want to CRISPR a segment in it to try and amplify X gene. And so you look inside of the plasmid and then you figure out, okay, like what part of the plasmid do I want to modify, or what gene? And so what you do is you take this and then you switch it to, instead of a circular, you can do a linear view, and it's easily obtainable through SnapGene Viewer. And then you can go to the specific sequence that you want to modify. And then you design primers forward and reverse around that to be able to amplify it or duplicate it or swap it out, which we'll learn much later in the course. But what reads it is those primers. And there are things like open reading frames, closed reading frames, origin of replication, which is right here in the yellow. And then you can see like there are some novel genes outside of the plasmid itself. So I can help you read that probably in the next tutorial. There's lots of things. I can't cover it all within an hour, but you can see there's lots of restriction digest enzymes that all flank the outside. So you can literally cut anywhere within the plasmid, within the circular plasmid, and say like, I want to insert like a gene like in this purpley area. Well, I can use the BAM-HI and the MLUL enzymes to like cut this segment out and then substitute it with the gene of interest that I'm amplifying or using. And then you would synthesize this complete genetically modified plasmid. So I'm not going to go over it today. We're going to go over just sequencing today, but I'll be sure to cover that in the future. All right, any other questions? JRD 210, GeneWiz doesn't really care where the sample comes from. I think it's just the cost. That's why their cost is so low. It's like, I believe it's like $7 to get it sequenced. So, yeah, I think it just might be the cost, really. Okay. It was great seeing you guys. Hope you have a great week. I'm gonna head out. But if you have any questions, just let me know. And I'll be sure to help you guys out. Okay? All right. Great. Thanks a lot, you guys. Take care. Bye.